Okay, so we're recording. Thank you, everybody. Welcome, everybody. This is our third and final event of the summer. So we started this last year. We had three excellent guests last year. This year we had two so far, and today we are hosting our third and final guest for the summer. So thank you very much for joining, and all of those that watch from through YouTube. We keep posting these activities there, these seminars, so everybody can see them and watch the presentation. Today, for the final guest of the season, is my privilege and honor to host today Dr. Supraja Varadarajan. I hope I said that right because I've been practicing. And Supraja is a young, new PI. She has her own lab for the last almost 2 years at the University of Texas in University of Texas Southwestern in Dallas, Texas. She is from Bangalore. She has a bachelor in engineering and biotechnology. She then had her PhD. She did her PhD at UCLA, University of California, Los Angeles, and then moved for a postdoc with Alex Huberman. Alex, right? Andrew, sorry. Andrew Huberman. I don't know where I said Alex. Andrew Huberman, very famous neuroscientist from Stanford University. She worked there since 2017 until moving to her new own lab at UT Southwestern. She has appointments in both neuroscience and ophthalmology and the O'Donnell Brain Institute. And she's really at this intersection between ophthalmology and neuroscience, looking at the regeneration of the optic nerve and using very, very cool methods that we will hear all about it. The title for today's talk is The Role of Retinal Recipient Neuronal Activity in Promoting Retinal Ganglion Cell Axon Regeneration. So I'll give you the floor. If anyone has questions during the lecture, please feel free to put them in the chat. And then at the end of the lecture, we're going to stop and ask Supraja all these questions from the chat, and whoever wants to interact with her and ask the questions directly. Okay, that's it. Thank you. I'm going to remove the share and allow you to how do I move this out? Stop share. There you go. There you go. Now you should be able to.
Let's see. I am trying to find out how to share, but in the meantime, thank you, Michael, for the kind introduction and for the invitation. I'm really excited to share my research today and give you a little snapshot of what my lab's been, my very new lab has been up to in the last year and a half. And thank you all for joining in. Again, if you have any questions, as Michael said, feel free to put it in the chat or also just unmute yourselves and interrupt me as we go. But I'm Supraja Varadarajan, and I've had a lab at UT Southwestern for about 1.5 years. And today I'll be talking to you about some of my postdoc work, which I'm now continuing in my own lab as well. So my lab mainly focuses on axon regeneration in the visual system. And we're particularly interested in how post-synaptic neuronal activity can be leveraged to promote regeneration, rewiring, and vision restoration. So these post-synaptic cells that I'm talking about, can you see my cursor? Okay. Yeah, so these postsynaptic cells that I'm talking about reside right here in the brain, in the subcortical region in the brain, just below the hippocampus. And they receive, these neurons receive visual input from the retina directly. And we're really interested in understanding if we lesion the retinal ganglion cells from the eye and increase neuronal activity in the brain, does that promote regeneration? Long story short, it does. I'll talk about how, and then we're exploring what mechanisms underlie this activity-induced repair. So I just want to remind everybody today that, we're obviously vision is a critical sense. You can see my slides, you can see me and Michael, you're using it. But vision is not just important for image-forming visual functions like object recognition and navigation. But it's also really important for non-image forming visual functions like sensing the luminance in your environment, how bright it is, which sets your circadian rhythm and regulates your hormones. It's also really important for image stabilization so that you can make sense of objects as you move around in space. And if you look at how our retina encodes all of this visual information and translates it to the brain, you'll see it really is a beautiful circuit, right? You have several retinal ganglion cells. There are many other types of neurons in the retina, as you may know, but today I'll be focusing on the retinal ganglion cells, the RGCs. And these are the only neurons in the eye that project their axons outside the brain, or to the brain. So these retinal ganglion cells encode visual information about these different visual features such as direction, contrast, luminance, and then relate that information to specific target nuclei in the brain, right? So that's how visual information about each of these features is encoded and then relayed to the brain. So for further processing through the V1 and the different cortical regions. Now, of course, when an injury or disease affects the visual system, it's debilitating. These axons disconnect from those targets in the brain. These axons degenerate, and it leads to vision loss. And there are many different conditions that cause blindness. Glaucoma is a major leading cause of blindness. It affects about more than 70 million people worldwide. And what I found really surprising is that there's at least 2.4 million eye injuries per year that occur in healthy adults between the age of 18 to 45, right? And then there are so many other eye injuries, optic neuropathies, and diseases that lead to blindness. So it really is an important, there is an important need to identify strategies that can restore vision and rewire these circuits to encode each of those visual features. Now, if we want to restore vision, then we need to achieve at least 3 major milestones. And these are the same milestones that a newborn retinal ganglion cell would need to achieve during development, right? So we need these retinal ganglion cells to survive. We need those cells to then regenerate their axons over long distances. And we need those regenerating axons to then reconnect with the correct synaptic partners. So they need to grow to the correct regions of the brain, identify the correct post-synaptic partners, form connections, become remyelinated in order to really restore visual functions. As you may know, mammalian central nervous system neurons, these RGCs are very much a part of the mammalian CNS, just like your brain and spinal cord. They really fail to regenerate spontaneously. And in general, our regenerative capacity reduces along with our immune competence as we age, right? So if you look at this little chart that's a little simplified, mature human adults have the least regenerative capacity. And so, this can be generally classified as cell intrinsic factors and cell extrinsic factors that contribute to a failure to regenerate. Cell intrinsic factors include lack of neurogenesis in adult mammals. There's a downregulation of growth promoters like mTORs and certain KLF factors. And there's an upregulation of growth suppressors like B10 and SOX3. Cell extrinsic factors in your environment are the glial scar that forms around a lesion or around degenerative debris. There's myelin-associated inhibition and prolonged inflammation, which also inhibits repair. Right. And so what people have done in the field for many decades now and have really, this is really fundamental work that has moved the field forward, is to use a very common model called the optic nerve crush, where you use forceps and crush the optic nerve just behind the eye, as shown here. And in some really foundational studies from Shiran Her's lab, we know that if you crush the optic nerve, where this asterisk represents the crush site, and you label these retinal ganglion cells, these axons degenerate back all the way to the point of lesion. So the retina is on the right and the brain is on the left, and the other way around. The retina is on the left and the brain is on the right, and these axons die back all the way to the point of lesion. But we know now from this foundational study, which is used as a gold standard, that if you manipulate cell intrinsic factors, and in this case, knock down P10, you can get these adult CNS neurons to regenerate their axons. But of course, since then, there have been many groups have done really phenomenal work and identified transcription factors, growth promoters, transplanted RGCs or modulated neural activity in the retina to really help us understand how to promote RGC survival and axon regeneration. And this list is in no ways comprehensive, and it's not even nearly updated. We have at least 4 or 5 really cool studies every year on these topics. But it unfortunately, we still don't really understand very much about how to get these surviving RGCs, these regenerating RGCs, to then reconnect with the correct postsynaptic partners to restore function. Moreover, all of the approaches that we know about so far are using presynaptic perturbation. What do I mean by that? We mean that if the RGC is damaged, then we're manipulating signals within the RGCs or their presynaptic inputs. But what really boggled me when I started my postdoc in Andrew Huberman's lab was what happens to the postsynaptic cells after injury. Right. And this is coming from because I was trained as a developmental neurobiologist, and we know during development in any system that target neurons or the future postsynaptic partners play a crucial role and provide important signals for formation of neural circuits. And this is the case in the visual system as well. We know that as these RGCs grow and extend their axons down the optic nerve and into the brain as the optic tract, they receive signals, axon guidance cues from the postsynaptic targets in the brain. This is their future postsynaptic partners that they manage to match up with. They receive trophic factor signaling from those retinorecipient neurons in the brain. And groups have shown that if you lesion these targets, ablate them, silence them, or deplete these signals, then it increases cell death and leads to incorrect wiring during development. So a really important point that I love driving home is that when RGCs form these visual circuits during development, they receive really critical signals from both the retina as well as from the subcortical targets in the brain. And yet we understand very little about what's happening to these postsynaptic targets in injury, right? And I particularly was interested in understanding if there was a way to leverage these signals, target-derived signals from these postsynaptic neurons to promote RGC axon regeneration and reconnectivity. But a problem at the time was that the most common model that has been used is the optic nerve crush model, which is a very severe injury, right? It leads to massive cell death, so that more than 80% of RGCs are dead within 2 weeks. And that means there isn't very much left to really repair, even if we try on many strategies. And no matter how powerful the repair strategy has been, we really cannot get enough axons to regenerate and to grow far enough into the brain. So we're at the point where we only get some axons to regenerate and restore partial functions. So that really doesn't leave us very much to work with if we want to understand what the role of postsynaptic neurons in repair is. So the first thing that I did during my postdoc was to use a different injury model called the distal axon injury model. And I'll walk through the visual system a little bit just because this is my favorite part. It's just such a beautiful system that is so tractable and easily accessible. But a very common strategy, as you may know, is that we can inject a tracer or an AV into the retina. In this case, I've used an anteroperate tracer called colorotoxin beta subunit, which is conjugated to a fluorophore. And if you inject that into the intravitreal space in the eye, it's very good at labeling the RGCs and their axons, so you can visualize the entire retinal fibril pathway as shown here. So these RGC axons extend down as the optic nerve out of the eye. In mice, more than 90% of RGCs cross the optic chiasm and extend as the optic tract in the brain, where they then send collaterals into 2 major targets, the lateral geniculate nucleus, or the LGN, and the superior colliculus. But what we're going to focus on is this little region called the pretectum, where these RGC axons also extend collaterals into several smaller targets in the pretectum. And so in the distal axon injury model, we use a scalpel to sever the axons anterior to the pretectum. And what we do is basically these axons they're basically extending past the DLGN and innervating the pretectum and the superior colliculus, and we're inducing a partial lesion on one side of the brain. So it's a unilateral partial lesion to the optic tract. And the first thing that we wanted to confirm with this injury was that cell survival and axon or axon innervation wasn't severely affected. So we took flat mount retinas, whole mount retinas from sham and control injury groups, stained them with RBPMS, which is a pan-RGC marker. And we noticed that 2 weeks after injury, there really wasn't any significant cell death. And this is important because we want these cells to survive and regenerate their axons so that we can understand or investigate the role of postsynaptic neurons. We also use CTB tracer to label RGC axons, and we noticed that we found that 2 weeks after injury, they still retained a lot of projections in the optic tract and innervations in the suprachiasmatic nucleus and the ventrolateral LGN, which is basically, these are all targets that are anterior to the point of lesion. So they're retaining some collaterals anterior to the point of lesion, and everything, or some of the axons after the posterior to the lesion are the ones that degenerate. So this was good. But as we know and can expect, these axons really don't regenerate. Lesioned axons really don't regenerate in the adult CNS. And this is an adult mouse, which is about 12 weeks old. So we really don't expect any regeneration here. And I do want to take a moment here to emphasize that this is a partial injury. And while it has its own caveats for specific questions, it's a very powerful model because it mimics clinical injuries in some ways better than the optic nerve crush, because we have some axons that are spared, so they're not lesioned. We have some axons that are lesioned and that do not regenerate, and then we have axons that are lesioned that do regenerate, right? And we can use two combinations of CTB injected before injury as a pre-injury label, and another CTB conjugated to a different fluorophore injected post-injury. And we can use these two labels to really dissect out unambiguously the degenerating, the spared, and the regenerating axons. So that's what we did. And I won't walk you through the details of this because this is all published work. But suffice to say that we can run these different tracers through an algorithm and pull out unambiguously the regenerating or the newly regrowing axons. And as I mentioned, because these are adult CNS axons, there's very little regeneration without treatment. So what we did was we wanted to modulate neuronal activity, and this is, of course, because neuronal activity is known to promote induced gene expression changes, both morphological as well as synaptic changes. And a previous paper published from the Huberman Lab has shown that modulating neuronal activity in the retina in the RGCs can promote axon regeneration in the optic nerve crush model. So neuronal activity was a very attractive approach. And so what we did was we induced the lesion, and then we used a chemogenetic approach to increase activity in the pretectums. These are the postsynaptic neurons. And then we used a pre-injury and post-injury tracer to identify the newly regrowing axons. And so this is what that image would look like, where the blue shows all the regions where neurons are expressing the HM3D chemogenetic protein, which can be activated by using a synthetic ligand like clozapine N oxide or CNO. And then the green and the yellow and the red are basically our degenerating, spared, and regenerating pixels that we can identify unambiguously. So I'll show you a little video here, which will basically help orient where in the brain the injury is, and in relation to that, where we are activating neurons and where the targets are. So this is CTB labeled RGC axons ascending as the optic tract, innervating the DLGN, pretectum, and the superior colliculus. And the blue is the region where we increase activity in the pretectum. And just anterior to that is where we induce the lesion. So again, the idea is to lesion these axons coming from the eye, increase activity in the postsynaptic neurons, and ask if those hyperactivated postsynaptic neurons can promote regeneration of these presynaptic lesioned RGC axons. And what we see is that, this is shown with the four, these are the controls. What we see is that when we induce a lesion to the optic tract and increase activity postsynaptically in the pretectum, distal to the lesion site, we see a robust regenerative response. And this, of course, is absent in the controls. So if we quantify that regeneration, we see that in controls, there is some regeneration. Remember, this is a partial lesion. So there are some axons that are spared. There are some axons that may be sprouting collaterals naturally. But overall, it's a very, very, very low degree of regeneration, if you can call it that. But when we increase activity distal to the lesion, we see a robust regenerative response. So the lesion site is where is what we're calling as zero. The blue bar is where is the region that we're increasing activity. So we're increasing activity about 800 microns away from the lesion site. And what we see is that in control, there's some regeneration up to about 200 to 400 microns. But when we increase activity, there's now a high degree of regenerating axons extending all the way up to 2 millimeters past the lesion site in 2 weeks, which is a huge degree of regeneration for central nervous system axons. But of course, a good measure of regeneration is how well these axons are able to reach their targets. And so we looked at each of these target regions, and this is there's too much going on here, but this is suffice to say that we looked at each of the targets, both anterior to the lesion, which is the DLGM, and all of the targets posterior to the lesion, so the OPM, the NOT, and the MDPPN are all small targets in the pretectum right here. And the superior colliculus is this major target in mice. About 90% of RGCs extend to the superior colliculus. And so if we compare our control injury animals and our animals that receive increased activity, we see a high degree of regenerating axons entering each of these targets in the group that had increased activity. So next we looked at how meaningful this regeneration is, and so I collaborated with Fei Wang, who's a postdoc in Xinduan's lab at UCSF, and we assayed the optomotor reflex, which is basically how well an animal can track a moving stimulus. So we can place a freely behaving mouse on a pedestal surrounded with screens with these drifting gratings, and the software can detect both the mouse's nose, head, and tail positions and tell us how well the animal is able to track the moving stimulus in the direction of motion. And this is a really good assay because we can, it's helpful since ours is a unilateral lesion, it's helpful for us to use the direction of motion in the direction lateral to the eye and assay the reflex. And so what we did was we collected a baseline response before injury, as each animal has a slightly variable threshold. And what the software does is it toggles between high spatial frequency and low spatial frequency of these these drifting gratings, and try to identify the threshold up to which mice can track that moving stimulus. So we collected a baseline response, and then roughly 2 weeks after injury, collected another post-injury response. And essentially what we see is if we normalize the pre-injury response to each animal's post-injury response, and the pre-injury is at 100%, and we see that the distal injury causes a deficit in this optomotor reflex in control mice. But increasing activity distal to the lesion allows these mice to retain the same threshold as before injury. So they're able to continue tracking the stimulus as well as they did before injury. And so this is really, this was very interesting to us. And it basically what it tells us is that we can increase neural activity distal to a lesion, and that promotes regeneration. It allows these regenerating axons to reach targets, and it rescues deficits in optomotor response induced by injury. But of course, everything that I've shown you so far is where I'm increasing activity broadly distal to the lesion. So this includes neurons that receive RGC input. So these are the bona fide retinal recipient neurons or post-synaptic neurons. But it also includes several other neurons in the vicinity that do not receive RGC input. And so I was really curious about whether we need to activate specific neurons, such as the post-synaptic neurons, or can any activity really help? And so I collaborated with Onkar Dande, a previous postdoc in the Huberman lab, and we focused on one specific target in the pretectum, which is the nucleus of the optic tract, or the NOT. And the NOT is a really cool target because it mediates one specific visual function, which is mediating horizontal eye movement. And so all the RGC projections that go to the NOT mediate horizontal eye movements. And so to focus on this target, we screened for a line and we identified this one synaptogmen 17 Cre line, which has Cre cells within the NOT. And so I first characterized this line by crossing the Cre line with an AN90 tomato. And we found that only in all the visual targets, the NOT was the only one that had Cre expressing cells. And so what this allows us to do is basically access these post-synaptic, bona fide post-synaptic retinal recipient neurons in one visual target. We further confirmed this using rabies tracing, where we injected a helper virus and the rabies virus into the NOT. And so any cells that are monosynaptically connected to the Cre cells in the NOT would be labeled in the retina. And in fact, we confirmed that there are many RGCs labeled in the retina, which basically tells us that these Cre cells in the NOT are receiving monosynaptic input from the RGCs. So in other words, this line allows us to manipulate postsynaptic neurons in the NOT. So we used this line and injected a Cre-dependent HM3D into the NOT. So now only those Cre-positive cells that receive visual input will have increased activity. And in controls, the NOT is outlined here in this dotted line. And in controls, of course, there's very little regeneration. But what was really surprising to me was that if we activate just a few hundred neurons in one visual target, it really promotes a robust regenerative response, which is quantified here. And these axons extend up to a millimeter past the lesion site. So of course, the degree of regeneration seen in activating one visual target is lower than activating something broadly. But what it really tells us is that activating postsynaptic neurons is sufficient to promote regeneration. And at this point, we were really curious about whether how much of this is true regeneration as opposed to, say, spared axons that are sprouting collaterals, which is a big problem in the field. And mechanistically, it's really interesting to understand whether lesioned axons are able to regenerate or whether these spared axons are able to make up for those lesioned axons. And so we used the rabies virus and injected that into the NOT and traced and looked at cells labeled in the retina. And so basically, if we are seeing spared axons that are sprouting collaterals, then both our control and our activity groups should have the same number of cells labeled. But in fact, what we see is that there is a threefold increase in the number of cells labeled in the retina. So what this tells us is that there may be cells that are sprouting spared axons that are sprouting collaterals, but there are also truly lesioned retinal ganglion cell axons that are able to regenerate when we increase activity in the NOT. And so basically, again, retinal recipient target activity, postsynaptic activity, is sufficient to promote regeneration of RGC axons. And of course, the million-dollar question is what is underlying this activity-induced repair? And so that's the mechanistic directions that my lab is heading in, which is really getting at, we see regeneration, activity is great to improve regeneration, but how is any of this happening? And so we have three directions in the lab, and I'll give you a little snapshot of those directions. But we're really interested in understanding what are the molecular mechanisms that underlie this activity-induced repair. So in other words, what gene expression changes are being induced by modulating neural activity in the postsynaptic neurons, and how is that promoting repair? But of course, this is a lesion site. We're increasing activity, and we know glial cells respond to both lesion as well as activity. And so we're really curious about how the neuron glia interactions are affected in the lesion site by this modulation of neuronal activity and how those contribute to repair or inhibit repair. And we're also interested in taking this approach into a degenerative disease like glaucoma, where degeneration happens slowly over time, to understand how to prevent degeneration. And so in our molecular mechanism, which is one of our immediate directions, we're really curious about what molecular signals are needed to rewire these visual circuits. And as I mentioned, during development, there are many different types of subtypes of RGCs that encode different visual features, and then relate that to distinct target nuclei. And so there's approximately 45 different subtypes of RGCs, and as a population, they map onto more than 40 visual target nuclei, as shown by this little schematic modeled after the London tear. And so really, if we want to restore vision, then this is the challenge that we're looking at. Like, how do we get these regenerating axons to really grow back to the correct synaptic partners and re-encode that same visual feature to mediate those visual functions? And so what we did was we collected, because we're activating these neurons, the virus has an M-cherry tag, we're collecting these hyperactivated neurons, hyperstimulated neurons. We sorted them for M-cherry, and we used three groups, where one is a sham group, which receives the virus but no injury. We have an injured group that receives the virus and a lesion, but no CNO, so they don't have increased activity. And then the activity group that receives a virus, it has a lesion and it receives CNO, so these neurons were hyperstimulated for 2 weeks. And so we did a bulk RNA sequencing to look at what these signals might be. And we're really interested in understanding whether these are growth-promoting trophic factors that are broadly promoting diffuse growth of axons that are lesioned, or are these more axon guidance cues where increasing neuronal activity really specifies specific signals in different targets, and RGC axons are responding with directed growth towards those target regions. And so I'm really excited to share this tiny little bit of new data that my summer intern, Hannah Bajaj, and research staff, Ben Chandy, in my lab have been working on. And what we see, of course, as you expect, is if we compare the sham and the injured group, then we see a host of gene expression changes. And if we compare the sham with the activity group, we also see a host of gene expression changes. But none of these are really meaningful because injury alone can induce a lot of gene expression changes. And so we compared the injury and the activity group. And to our surprise, we see fewer gene expression changes when we compare injury and activity. But of course, when we look at the signaling pathways that are enriched, we see that all the right signals are signaling pathways are enriched. The cell migration, cell differentiation, growth, and really signaling pathways involved in axon extension and guidance are upregulated. And so right now, we're really interested in understanding what exactly these molecular signals are and which genes are involved and what their functions are. And so to basically get an understanding of what does increasing neuronal activity do at a molecular level to promote this axon regeneration and rescue those oculomotor deficits. But in the second angle, what was really surprising to us is that we see a huge change in the number of genes that are enriched in the population outside the stimulated neurons. So the stimulated neurons are the postsynaptic neurons that receive the virus. But remember, there are many other neurons in the vicinity, many other cells in the vicinity that also that are not activated, that are not hyperactivated by the chemogenic dread virus. These could be other neurons, or it could be glial cells. And so we collected those cells as well. And what we see, to our surprise, is that there's almost twofold increase in the number of genes that are enriched compared to the postsynaptic neurons that are hyperstimulated. And so we're really interested in identifying whether these gene expression changes are affecting glial cells, oligodendrocytes, astrocytes, and microglia, all of which are known to respond to both the lesion as well as changes in neuronal activity. And so we want to tease apart what the contribution of each of these cells are towards tissue repair and reconnectivity. And with that, I will summarize. And today, basically, what I've shown you is that we have this distal injury model that we can use to get at the role of these postsynaptic neurons and target-derived signals in repair. I've also shown you that increasing activity broadly distal to the lesion site can promote axon regeneration. And I've shown you that increasing activity specifically in postsynaptic retinorecipient neurons is sufficient to promote regeneration. So both of those really speak to the role of neuronal activity post-injury and in repair. And it gets us thinking about whether we want to maintain the level of activity or perhaps enhance activity after injury to promote better repair. And what is really exciting is that this strategy of tapping into postsynaptic activity and target-derived signals can really be used to treat many eye diseases and optic neuropathies like glaucoma, optic atrophy, and other optic neuropathies, but also in general to many other CNS injuries and diseases like traumatic brain injury and spinal cord injury. And especially with non-invasive brain stimulation becoming more common as a method and treatment to treat stroke and epilepsy and other degenerative conditions, it's really important to understand what are the mechanisms underlying neuronal activity-induced repair. With that, I will thank all of you for listening and Michael for this opportunity and everybody who's contributed to the previous work, like Onkar Dande, Famli, who is my undergrad in the Huberman Lab, and Fei Wang and Shintuan's lab. And I will thank my lab and our funding sources as well. And I'm happy to take any questions.
Excellent. Thank you so much. Thank you. Thank you so much. This was really, really interesting. Let's open the floor for questions. Anyone in the audience that wants to ask anything? I have a bunch of questions, but I'm gonna give the right of way to others. No? Nobody? Okay. So I have a bunch of questions. The first one is an anatomical question that exposes how ignorant I am about this, but I don't care. If your lesion is before the tectum, before the pretectum, right? Are those axons also the ones that continue to the superior colliculus too? Or can you differentiate between those that end in the tectum and those that continue to the superior colliculus?
No, so those are the ones that most likely are also continuing to the superior colliculus, because at least in mice, the superior colliculus receives input from 90% of RGCs.
Right.
So it's really hard to distinguish just the 10%, but so most likely the axons that are lesioned they have a collateral going into the pretectum and certainly ending in the superior colliculus, or yeah.
So in theory, if you express your dread in superior colliculus and you increase activity there, you should also, in theory, promote regeneration to that area, right?
Yeah, I mean, actually, we see yeah, two things. I think one is, yes, that should be the case. And in our experiments, we have actually seen axons regenerating all the way till the SC and into the SC as well, which is past the lesion site. So it's not that if we stimulate one region, they only extend up until there, they extend to beyond the lesion, the activation site as well. And the second was that Liev Moon's group in Belgium has actually done some really cool studies using glaucoma models where they optogenetically stimulate the SC. And but that was looking more at degeneration in glaucoma, and they do see a modest neuroprotection of RGC survival.
Okay. So this is the type of technical question that is extremely annoying, but I cannot
No, I love it
myself,
for sure.
and I have to ask it. I mean, you're expressing dreads, you're hyperactivating the NOT or whatever area. How much? How big is this activity as compared to the normal activity?
Yeah, I mean, they are, it's with dreads, right? So this is not trying to mimic physiological activity or double physiological activity. They're hyperstimulated. They're firing like crazy for a period of, I think once you inject CNO intraperitoneally, the activity peaks around 90 minutes, 60 to 90 minutes. And then the half-life of CNO is about 4 to 6 hours. And then we give them injections twice a day. So we're really trying to go for like a 10 or 12-hour stimulation based on the previous studies have shown. So it's not trying to go for a physiological level, but I would be really interested in understanding how it compares to physiological activities, twofold, threefold.
Right. So you don't have measurements of, hertz, firing frequency.
No.
Okay.
No,
And
but I'd love to talk to you about how to do that.
Yeah, sure. And now the next question is another question that I hate, which is the experiment that you didn't do. You can never answer a question about the experiment that you didn't do, but we can speculate, okay? We can run wild with our imagination. So if you are hyperactivating the superior colliculus or the NOT or the LGN, whatever, to attract the ganglions that have died, the ganglion cells, the axons of the ganglion cells that are dying, but you're still hyperactivating this guy who is connecting to its post-synaptic target in the visual cortex or whatever. So what is happening to them? Are they changing because hyperactivity of the middleman in this pathway?
Yeah, I don't know. That's a really good question. It's really, I mean, obviously when we're looking at visual function, it becomes really important to understand what is happening to the cortex because of the stimulation. But I think, at the end of the day, if those retinal ganglion cell axons are not connecting, or no longer connected, then there is no vision,
Right.
right? So regardless of what it does to the V1. I mean, of course, if in thinking of this translationally, it becomes important to understand how to modulate this to a what is the permissible limit to which we can modulate without having debilitating effects on the V1. But I'm actually really curious on if it provides any neuroprotection to those postsynaptic target neurons, and because the ones that lose their RGC input will maybe eventually start to die, maybe not because they're interneurons, but what does the activity help them survive better? And then, of course, yeah, I mean, like local circuits, what happens to local circuits when we hyperactivate these neurons? We really haven't gotten to any of that.
Right. So
Would
actually,
love
one
to.
of our guests here wrote a question that really, is the continuation of that. I have a question. What happened when you stimulate the visual cortex? So what if you did I assume the question is, what if you did your lesion in the same place, but you stimulated the cortex after the second synapse and not at the first synapse?
Yeah, no, that's a really cool question too. I haven't considered that. But in terms of if I may, I think what the question is getting at is how far away from lesion is this helpful? And so we're doing some experiments to do the same thing in basically an optic nerve brush. So if we move the lesion site further away but continue stimulating the same neurons, is that still beneficial or not? And I don't know distance-wise if that's the same as to the visual cortex from the pretectum. But yeah, I mean, that's a really good question to ask as well.
So
I would, yeah.
no, keep going.
Yeah.
Sorry.
No, I would just imagine that might be even more complicated because we're looking at the postsynaptic neurons that presumably don't get injured, right? And because we're stimulating the cortex, whereas we're lesioning the retinal ganglion cells. And so that now we're talking about one step further away.
Right. But I think we have evidence of the opposite. So there are studies. There was just one study published not that long ago that showed that when you have a cortical injury, you see death of ganglion cells. So there is retrograde death of ganglion cells backwards when the injury is originally in the, like cortical blindness. So people look
Right.
at cortical blindness, they think, okay, the whole system is great. It's just the cortex that is now blind. But actually, no, it's not just that. There is retrograde induction of degeneration all the way back to the retina, which is amazing.
Oh, interesting. I did
Yeah,
not know that. But yeah, I guess in that case, in those kind of conditions, stimulating these neurons may be beneficial,
right,
and prevent RGC death
right.
potentially. But
Well, people
that'll be cool to look at.
that have visual impairment because of stroke, I mean, they
Yeah,
do rehabilitation by looking at images and retraining the visual system. So they are
right,
overactivating themselves, right? I think that
right.
also that was part of what you wanted to get to. That, I
Yeah,
mean, maybe with a magnet or maybe with
yeah,
visual training, we can activate the right. I mean, coming from the eye, of course, it's going to be a problem because you have an injury there. But if the injury is unilateral and you still
right.
the second eye, would that work? Do you think the information will cross hemispheres and help you one good eye repair the bad eye?
I don't think, I mean, in mice, that's really hard, right? Because they only receive 10%, but perhaps in humans, that might be a better question to investigate. Yeah,
Awesome, awesome. Thank you so much. This is fascinating.
of
This is really
course.
fascinating. I really want to know what's the end of the story. Like, what did you find? What is the, you set up the whole thing and then you didn't
Yeah,
give us the end of the movie. We have to come to for the sequel. We have to come to
yes.
the second
Yeah,
part.
in a
Yes.
couple
Okay.
years, yes.
Okay.
Thank you,
So I
Michael. Really
assume you're
appreciate it.
you're targeting some neurotrophic factors or some maybe extracellular matrix proteins that help the axons guide
Yeah.
themselves or
Yeah, basically, I mean, we're not limited to anything specific. My jam is axon guidance. So, of course, I'm trying to look for like how do these axons get to where, because the remarkable thing that we see is that these axons really don't stray out of the optic tract and wander anywhere that they're not supposed to. They do still stay in the optic tract. They go only to the regions that they're supposed to go to, which kind of indicates there may be some directive information. But that could be from many other sources too, because it is a partial lesion. So we're trying to get at some of those aspects as well.
But you're basically awakening developmental plasticity that is supposed to be dormant in these mice because they are too old, they are adults. And
Yeah,
basically by activating these neurons, you are basically recapitulating that developmental plasticity that we used to have when we were embryos or very young
right.
in life. That is
Yeah,
fascinating.
exactly. And that's been shown in, I mean, in so many contexts, right? Like not just visual injuries, but also spinal cord injury, that is possible. You can develop, you can recapitulate developmental mechanisms. But I think what is cool here is that we're not looking at the injured neurons, but something else that's external to that neuron. It's a downstream partner, and that can provide beneficial information as well,
Right.
which has not been looked at except in development. So
Awesome. Very, very nice. Thank you so much, Supraya. Thank you everybody
thank
for
you.
joining. There is, oh no, it's another person saying thank you. Everybody, thank you for joining. This will be on YouTube for everybody to watch. And that's it. We are done with the summer series. Thank you, Supraya. Thank you everybody for joining us. We'll be in touch.
Thank you,
Bye,
Michael.
everybody. Bye
Appreciate it.
bye.